Alrighty, everyone. So to get started for today, we are going to do a quick review of what independent assortment is. So we start off with the question, do the alleles of one gene control how the alleles of another gene are expressed? For example, one thing you could think about is, do you think that the genes for our eye color would somehow control the genes that control our hair color? And if you think about that for a second, the answer you should probably come to is probably not. And that's we kind of call that idea independent assortment. This is, in most cases, genes will not affect how other genes are expressed. So when we're thinking about this, in that example, hair color would not affect your eye color. This can explain how siblings with the same parents can show different traits. So for example, me, I have brown hair and blue eyes, but my two brothers, they also have brown hair, but one of them has green eyes and the other one has brown eyes. So this can show that through the idea of independent assortment, just because one gene gets passed down from parent to offspring does not mean that it's going to be the same set of genes every time. All of these genes are going to be sorted independently so that we can allow for a larger variation within individuals. And when we think about evaluating traits all at once, just because these two genes are not connected, it does not mean we can't evaluate two genes at the same time. When we think about what we've been doing so far in genetics, we've been doing a lot of what are called monohybrid crosses. And this would be a genetic test to evaluate one trait at a time. So again, this would be an example where we're just looking at hair color or just looking at eye color. However, we can do a different type of cross called a dihybrid cross. And this is going to be a genetic test where we evaluate two traits at once. And to just compare and contrast these two a little bit, I just put up a Punnett Square, and this should look pretty familiar. This should look pretty comfortable for us. We've done these types of crosses before. We're just looking at one letter. We see that the yellow allele is going to be dominant to the green seed color allele, and that when we have that heterozygous genotype in the first top two boxes here, we have our yellow seed pod being shown, and then in the bottom, when we have a homozygous recessive genotype, we're going to have this recessive trait being shown, which in this case is this green seed color. But when we move on to dihybrid crosses, things get a little bit more complex. So first, take a second, go ahead and compare these two Punnett squares and try to come up with a few differences. You can even pause the video. So if you take a second to compare and contrast these, what we should hopefully notice is a couple of things. First off, just the general size is very different. So in this first Punnett square, we have 4 boxes. This means that we are predicting the probability of 4 offspring being formed. However, when we move into a dihybrid cross in this bottom picture here, we now go to 16 boxes. We basically have 4 times the amount of predicted offspring when we're thinking about a dihybrid cross. Again, dihybrid cross being a genetic test evaluating 2 traits. Another thing that you should notice is when we are looking on the outside of our boxes here, we'll notice that there's only Ys in a monohybrid cross. But because we are now dealing with 2 traits, we now need to use 2 different letters at once. So in this case, we are evaluating the genes controlled by, we're going to say, with an R and a Y. And when we combine these together, when we usually have 2 letters per box in a monohybrid cross, we now have 4 letters per box in a dihybrid cross. So there's a couple of differences here. And what we're going to start to do now is to think about how do we even come up with these letters on the outside in order to even set up our dihybrid cross. Because you may be able to see how these letters get distributed pretty easily, but we're going to talk a lot about how are we going to set up this dihybrid cross? How are we going to get this ready to work with?